Hello, everyone. Today, I am here with Monica Sandoval, and my name is Nadia Mitra, and we are here with Dr. Frye. Monica, if you can take away the introduction.
Absolutely. Dr. Richard Frye is a child neurologist with expertise in neurodevelopmental and neurometabolic disorders. He's fellowship trained in behavioral neurology and psychology. He has a broad background, including training in physiology, psychology, and biostatistics. He has authored over 150 peer-reviewed publications and book chapters and has conducted multiple clinical trials. Dr. Frye is the chief of the Division of Neurodevelopmental Disorders at Phoenix Children's Hospital and the director of their autism program. He has helped thousands of families, and we are honored to have you here today.
Thank you so much. Thank you for that introduction. I'm really excited to be talking today to everybody, and excited about the interest and to tell you some great information that I hope will help children.
Yeah, so Dr. Frye, just a little bit about ourselves here at the Autism Research Coalition. We're a group of clinicians, researchers, parents that are very vested in autism research, and we are privileged to have you here today with us. You are an expert in mitochondrial dysfunction, folate pathways, nutritional deficiencies, PANS, PANDAS, you name it. There's no stone unturned. And we talk about warrior moms, warrior dads, and to us, you are a warrior researcher. And so to begin with, I'm just hoping you can kind of explain to us mitochondrial dysfunction. So my first question for you is, can you explain in layman's terms, what is the mitochondria, what is ATP, and what is the electron chain pathway?
Sure. Do you want to share my slides? And I have a kind of picture here for you. Can you see that? So the mitochondria is what we call a subcellular organelle. That is, it's a part of the cell. So as you know, our body is made out of cells. And all of our cells, in order for them to operate, they really need energy. And one of the places, the main places that the cells get energy is from the mitochondria. And you can see in this picture that the mitochondria is inside the cell, and if we blow it up, it's very complex. It's a very complex part of the cell. And of course, to start with, we should know that that we don't just have one mitochondria in our cell. We have anywhere from hundreds of mitochondria to tens of thousands of mitochondria in each cell, depending on how much energy the cell needs. So skin cells may only have 100 mitochondria, whereas muscle cells that need a lot of energy may have tens of thousands of mitochondria. Other parts of our body that need a lot of energy, like the brain, has lots of mitochondria. The nerve cells have mitochondria in key places where energy is needed. So the mitochondria is really key for our cells, those building blocks of our body, to function. And what we know is that the mitochondria is very sensitive to many different things and many different stresses. So the mitochondria can be stressed very easily. And so we worry about that in many diseases. And we know that the mitochondria, we're learning that the mitochondria is involved in many different diseases. Maybe not so much as a primary disorder, that is a disorder where the disease starts in the mitochondria, but sometimes as a secondary disorder. That is because you may have some type of disorder, especially inflammatory disorders, cause so much stress on the body that they tend to push down the ability of the mitochondria to function. So quickly and kind of in layman's terms, how does the mitochondria function? So the mitochondria takes certain fuels. It really likes carbohydrates the best, but it also breaks down fats, and then it also can break down amino acids if it has to. Amino acids are the building blocks of our proteins that make our body. And these nutrients come into the mitochondria, and through a series of steps of biochemical reactions, they get turned into what we would think of the energy currency of the cell, kind of like the Bitcoin of the cell, is something called ATP. So that's the currency of the cell, and the ATP can be used by other parts of the cell as energy. There's a number of different key biochemical reactions that turn the fuels of our body into ATP. And the two most important ones that are pretty well known is something called the Krebs cycle, which is a cycle that creates something called electron carriers. And those are molecules in the cell that take electron energy from the Krebs cycle and bring it to the final common pathway where you make ATP, called the electron transport chain. And the electron transport chain is where many of these biochemical reactions converge and allow the production of the ATP molecule that's so important. And if you can see in this diagram that the mitochondria on the top right there is a very complicated organelle. It has two layers. It has an outer layer or membrane, and then it has this inner membrane. And the inner membrane is very important because it has these folds in it called cristae. And those are very important because that's where this electron transport chain sits, right in those cristae. And you can see that inside the inner membrane is something called the matrix, and the matrix is where lots of other stuff happens, lots of other important stuff. One of the unique things about the mitochondria is it's the only other part of the cell besides the nucleus with DNA. DNA, of course, are the instructions that tell our cells how to work. We always think that our DNA is on our chromosomes, which are located inside the nucleus. And for the most part, that's very true. And our chromosomes contain about 23,000 different genes that control our cells, including about 1,500 to 2,000 that control the mitochondria. However, the mitochondria itself has some DNA. It has some instructions that are contained within it, which code for about 60 or so different proteins. And it also controls, how the mitochondria works. So this makes something called inheritance, when we look at inheritance and how diseases are inherited, very complicated, because your mitochondria are inherited from your mother, whereas the genes in your nucleus are inherited from both your mother and father. In addition, this mitochondrial DNA, you have several different copies of it in each mitochondria. And so what happens is that sometimes not all those copies have the right code on them. And so we can find mutations and abnormalities in some of the copies and not in others. This is so-called heteroplasmy, which makes, of course, inheritance and finding these genes that are abnormal somewhat more difficult and complicated. But the other thing that's really important about mitochondrial DNA is that it's not as resilient as the DNA in the nucleus to certain types of stresses and toxicants. So you can get acquired deficiencies in mitochondrial DNA and mutations because of ongoing and repeated times of stress. So the mitochondria is important and not only in giving energy to the cell, but also it has other biochemical pathways that support other parts of the cell. And if you look at a diagram of all the biochemical pathways in your cell, the mitochondria is right at the center. So it tells you how important this one piece of the cell is for the cell to work. And so in that sense, it's important to know that dysfunction or problems with the mitochondria can affect many parts of the cell, and there's many different inroads of how the mitochondria can be affected adversely. So we're not surprised that we find it involved in many diseases, including autism.
Okay. Yeah. So what are some things that we could see creating mitochondrial dysfunction? Would it be reactive oxygenative species, environmental toxins, illness? What sort of factors do you see causing issues in the mitochondria?
You're absolutely right. The mitochondria can be stressed by many different factors, and many times we think of things like external toxins, that come in and hurt the mitochondria, and that's definitely true. That can happen. So toxins from the environment can stress the mitochondria, but we're also finding not having the right nutrients can actually stress the mitochondria, because the mitochondria needs certain nutrients and cofactors to function. And if it doesn't have those, then it will not function properly. Oxidative stress caused by other metabolic processes are very important. The mitochondria actually produces a lot of oxidative or reactive oxygen species because of all the biochemical reactions that are ongoing in it. And reactive oxygen species themselves can be deleterious and can cause damage if they're uncontrolled. That's how we get oxidative stress. So if the mitochondria itself, if it does not control the reactive oxygen species it makes, it could actually damage itself. Reactive oxygen species are also caused by other things in the body, most notably inflammation. So when you have inflammation, you many times have high levels of oxidative stress, and if that is not controlled, that can damage the mitochondria. And then we know that there's certain types of genetic disorders that seem to predispose some dysregulation of the mitochondria. And some of the things that we're finding, which is very interesting, is that it may not be just the mitochondria itself being damaged, but we also have systems in the cell that replenish the mitochondria, restore it, and make sure that it's working optimally. And we're finding that there's many disorders in where these systems that really try and keep the mitochondria fresh, so to speak, are not functioning. And so the mitochondria get worn out pretty quickly and can't function as optimally as we'd like.
Sure. So if you had to give us a percentage of children that have mitochondrial dysfunction in the autism category, how would you describe that? And how may they present? Would you see a regression in language? Is it something that is there at birth, or is there a later onset? If we were
Sure. And so so briefly, so for what type of symptoms we see for mitochondrial disorders, one of the things we think about, and I'd like to contrast mitochondrial disease from mitochondrial dysfunction. And so there's many types of mitochondrial diseases, and this is when there's a primary problem with the mitochondria. And really, the first types of mitochondrial diseases were described in about 1988, which is in the span of medicine. That's pretty short time span. So it's not surprising that we don't know a lot about the mitochondria at this point. We're still just learning about mitochondria and mitochondrial diseases. But what we find in those diseases is that usually it affects what we call the high energy dependent tissues. That is the nervous system, the brain and peripheral nerves, the GI system, and the immune system. And as we know, these are three areas that we see a lot of problems with in kiddos with autism. So it's not surprising that the mitochondria may be involved. So how many kids may be affected by this? And so we really set off to ask this question probably about 10 years ago now. Myself and Dr. Rossignol said, well, we've been hearing that there's people talking about mitochondrial disease and autism. What is the evidence for this when we look at the medical literature? And so in this paper we published in 2012, which was something called a systematic review and meta-analysis, we looked at every paper that was published on mitochondrial problems and autism, and we compiled the data. Now, I'll just, not to get overly technical, but I'll just talk a little bit about what our findings were. What we found is that the three pretty well done studies that looked at the prevalence of mitochondrial disease and autism put it at about 5%. Okay. And this is, in the terms of mitochondrial disease, this is really a big number, because mitochondrial disease in the general population is less than 0.1%. It's a rare disease. So 5% of kids with autism having mitochondrial disease is a big number. However, that's a small proportion of kiddos with autism. But you have to understand that mitochondrial disease is, to make that diagnosis, there's very particular criteria you have to meet. So what we also did is we asked, well, what do the studies show for what we call biomarkers of mitochondrial dysfunction? That is kind of blood tests that tell us that the mitochondria really isn't working as well as it should. And when we looked at things like increased lactic acidosis, we found there were 6 studies that showed that the prevalence in autism was about 30%. So that's much bigger than 5%. And that some of these other markers, pyruvate, lactate pyruvate and such, also had from some large percentages. So we thought, well, maybe there's some discrepancy there. Maybe there's some kids with mitochondrial dysfunction that might not have mitochondrial disease. And then I'll show you this slide here. This is a study from UC Davis, the Mind Institute, where they looked at immune cells of kiddos with autism and compared them to age and gender matched controls, and asked how many of those 10 kids seemed to have some type of problem with mitochondrial function. And they found it was 80%. They found that it was 8 out of 10. So 8 out of 10 is much bigger than 5. 80% is much bigger than 5, and actually much larger than the biomarkers that we saw, which seemed to be abnormal about 30 or 50% of the time. So really, this is something that we're really still, looking into, and we're starting to recognize new types of mitochondrial dysfunction that might not fall into this mitochondrial disease category. However, going back to mitochondrial disease, we want to ask, well, are there specific symptoms that we see in kids with autism with mitochondrial disease? And the answer is yes. We reviewed every paper of children that were diagnosed with autism and mitochondrial disease and asked, are there specific characteristics that separate them from the general autism population? And we found out that yes, there is. And some of this is a little bit interesting and surprising. So first of all, we found that that they tended to have seizures. That about 40% of them had seizures, so they were more likely to have seizures. And they were more likely to have GI abnormalities. So 74% of them had GI abnormalities. So those are 2 clinical characteristics. But one of the other things that was really interesting is we found out that more of them had neurodevelopmental regression. So regressing from being typically developing to having autism suddenly, but also more of them had motor delays. So that is that their gross motor development was delayed early on. And those may seem to be at odds with each other, normal developing and then having this regression, and then those that have early onset delays. But it actually seems like there's these 2 subsets. So one of the red flags is neurodevelopmental regression, having normal development and then suddenly losing it. And we know from other studies that certain triggers such as fever or infection or any type of possible immune challenge may be associated with neurodevelopmental regression associated with mitochondrial disease and autism. Also, we see that there's a group with motor delays. So a lot of children with autism don't have gross motor delays. They walk and run and do all of that fine. They may have some apraxia, some coordination issues, but as far as gross motor function, they don't have any early delays. And then there's some that do. And so this is a big red flag for the kids that may have mitochondrial disease very early on and start early in life. So there seems to be these two different kind of subgroups. One of the other things we found is when we look to see how many of these kids with very, critically diagnosed mitochondrial disease had DNA abnormalities, mutations in the DNA to explain their mitochondrial disease. It was only 23%, suggesting that the great majority of them did not have kind of classic DNA mutations to explain their mitochondrial disease. Thinking of other types of complicated factors, including environmental factors that could have affected these children to cause this is a real possibility.
Okay, so if a child presents themselves to your clinic and they're showing signs of fine motor delay or they're having seizures, you have an algorithm, a treatment algorithm that I saw published just based on how you would assess for mitochondrial dysfunction and then how you would treat it. So what sort of biomarkers do you look at in clinical practice to identify these children that have mitochondrial dysfunction? And how would we suggest this to our providers to look for this based on like serum levels? Do you suggest a mito swab, OAT test? What sort of labs would you recommend? And then based on those labs, what specific treatments do you recommend based on those results?
It's a good question. It's a complicated question. One of the problems is that a lot of these markers are not so straightforward to interpret. So I start out with these biomarkers of mitochondrial dysfunction, fasting labs. I think it's always important. One of the biggest problems I see when I get labs from outside is that they weren't done fasting first thing in the morning. And the reason why we do that is because we know that the metabolic systems of your body are under the greatest stress after you've had some type of fast. The natural time that you fast is when you go to sleep. You can't eat. You can't snack. It gives us an opportunity to see the body at its maximal stress. And this brings out these markers, so it's a little bit more sensitive. I always recommend labs first thing in the morning to look at mitochondrial function, and there's a whole list of different things I look at. Lactate and pyruvate are the classic markers. Amino acids are very important. Then it's important to look at the fatty acid oxidation pathway. So it's important to look at carnitine and acylcarnitines to see if there's a problem with the fatty acid oxidation pathway, which we see in some kiddos with autism. I always check homocysteine because we know that many children have problems with methylation and oxidative stress, and homocysteine is something you can order in a clinical lab that may give you an indication if the methylation pathway may be under stress. And then I also do some key vitamin levels too, especially kiddos that are not eating well and have food restrictions, because there's some easy stuff you can do by just replenishing vitamins that can be low, including vitamin D, which we know is so important, CoQ10, of course, looking at zinc and copper. So that's where I start to look for any type of signs of mitochondrial dysfunction. And then if I see anything or I'm highly clinically suspicious, yes, I do move on to the mito swab, which allows us to swab the inside of the mouth and then measure several of the enzymes in the mitochondria. And, we did one study, which is here, where we looked at our clinic where we've used the mito swab. And we found that, and of course, you have to understand how to interpret that too, because usually classic mitochondrial disease is associated with these enzymes not working very well, below normal. And you can see on this graph, this is a study we published of our patients in our clinic. And what we did is we looked at their mitochondrial function. And in this graph, you can see that we measured 3 different enzymes, which you get with the mito swab, and that all the black dots are within normal range, and all the red dots are outside the normal range. And we can see that there's some red dots that are below where they should be, below the normal range. And that's kind of consistent with more classic mitochondrial dysfunction, that the mitochondria is not working, well enough. However, you can see that there's red dots really above that normal range too, in all of the enzymes. And that's significant. And what we found is really that sometimes we find that the mitochondria is working a little too hard. And that may not be good for it. In fact, we find that when it's in that state, it's very vulnerable to stresses, and may be associated with neurodevelopmental regression. So you have to be a little bit knowledgeable in how to interpret these things. But we do go on to the mito swab when we suspect that there might be mitochondrial dysfunction, although I don't do it initially because I think you really need the whole picture to interpret, to treat the child. And then we do move on to treatment. And one of the controversies there is, many people consider that really you should only treat mitochondrial problems if there's what we call primary mitochondrial disease. That is a primary problem with the mitochondria. But in this paper that we wrote, myself and Dr. Kaler and Dr. Nazoff, we argue that that's probably not optimal. That even if there's secondary mitochondrial dysfunction, that is, if you find that the mitochondria isn't working well, and even if you don't know why, it may be an advantage to really treat the mitochondrial dysfunction. And in this paper, we actually give several examples of diseases, genetic diseases, where there's clearly the gene is clearly not a mitochondrial gene, such as muscular dystrophy. That doesn't have the genetic defect doesn't have anything to do with the mitochondria. But what you find in muscular dystrophy is because of the inflammation and the problems with the muscle that develop, it results in secondary mitochondrial dysfunction, and that many children will respond to treatments, even though that's not the primary problem that they have. So we really think that if there's signs of mitochondrial dysfunction, you should at least give some try of really treating it. How would you treat it? Well, we have this thing which we call the mitochondrial cocktail, and that and that's just kind of a broad way of saying that we want to give support to the mitochondria, using different cofactors that will be important for the mitochondria to function. And here in this table, we just give a list of all these things. Of course, one of the most important things I always start with is CoQ10, usually in the form of ubiquinol. CoQ10 is an antioxidant, and people think, it's a cofactor. It's probably important for the mitochondria to function, but it's actually much more than that. CoQ10 works at that electron transport chain I was talking about. And it actually carries the electrons from the complexes, between the complexes. So you can think of it as kind of the wires of the electron transport chain. So it's really important. If you don't have it, it's like you don't have those wires to connect the different parts of the electron transport chain. Of course, ubiquinol is the reduced form, the more bioavailable form, and I always encourage people to use that as the first treatment in the type of CoQ10. Then we have what we call more of the mitochondrial cocktail, which are B vitamins, mostly, that help the mitochondria work. So all the machinery, all the enzymes inside of the mitochondria need these cofactors to work. So niacin and riboflavin, biotin, thiamine, pantothenic acid, pyridoxine, and alpha-lipoic acid are all very important. And that's what's usually contained as the starting of a mitochondrial cocktail. So then I usually use a multivitamin that has a lot of these in there that are made to support the mitochondria. And then on top of that, the other really important thing is carnitine. Carnitine is something that we see low in kiddos with autism, just in general. We know that one of its major purposes is for fatty acid oxidation, that is helping you to burn fats to make energy, but it also has another purpose. It's really good at getting rid of toxic organic acids that are made because the mitochondria and other biochemical pathways aren't working correctly. So it's a great scavenger for these and really helps get rid of those toxic elements. So what else? So then really we want to support the other systems that are involved with mitochondrial function. And if you look at the bottom, we have redox metabolism support. And so it's very important, as we talked about, that oxidative stress can cause problems with the mitochondria. So we want to reduce that as much as possible. And my preferred way is really using a methylcobalamin and a type of reduced folate, usually folinic acid, in combination to promote the production of glutathione, which is the major antioxidant in your body and in the mitochondria. This has other advantages. I don't usually give glutathione directly because I believe it's important to push the body to make glutathione for a number of reasons. Another thing that you can use is N-acetylcysteine, which will also help with the production of glutathione. B12 and folate not only may help with the production of glutathione, but may help with some of the redox, some of the methylation abnormalities also. So it may have another advantage. And then something I might talk about if we have time is the support for folate metabolism. And one of the things that we know is that in many kiddos with autism, folate is not getting into the brain. And there's a number of reasons. And one of them can be because the mitochondria isn't working all that well. And so we want to make sure that we give something like Leucovora and calcium at high doses to push folate into the brain so it functions better.
Okay, yeah, so just to kind of summarize, some mainstays of treating mitochondrial dysfunction would be CoQ10, some B vitamins, carnitine, and then ways to address oxidative stress along with supporting the folate pathway. And then on the topic of carnitine, is there a version of carnitine that you recommend over others? That's been a common question that's been coming up. And are there any disadvantages to taking these supplements or contraindications? Would they feed bad bacteria, for example? That's been a common theory among families and providers as well. So can you share a little bit about that?
Yeah, no, I mean, it's a good point, especially with reference to carnitine. So So I get the question, is acetyl L-carnitine or L-carnitine the preferred carnitine? So the studies, we have 2 double-blind placebo-controlled studies on carnitine and autism, both of them positive, and they used L-carnitine. Other studies on other disorders have used acetyl L-carnitine for neuroprotection, but they haven't used L-carnitine. So there's no real head-to-head comparison. There should be no difference between acetyl L-carnitine and L-carnitine as far as the metabolic effects of it. And one thing I should say with taking any vitamins and supplements and even medications is that reactions to medications and supplements and such many times are because of the additives, not because of the active ingredient. So it's important if you have a reaction to something to maybe try a different brand that you may not react to. So I think you have to kind of temper some of the reactions you get to these things with knowing what the additives are. But with that said, there is this kind of controversy with carnitine. I think it came out of an interesting study that suggested that carnitine can increase the risk of cardiovascular disease in adults. And when they really look back at that study, and they found that, well, no, it wasn't the carnitine, it was the red meat eaters. So if you didn't eat red meat, if you were vegetarian, carnitine didn't increase your risk of cardiovascular problems. But if you ate red meat with lots of really bad fats and took carnitine, which transports fats very rapidly, then all those bad fats were being brought into your body. So in the sense, it wasn't carnitine, it was the diet. In the same sense, sure, if you have carnitine's great at transporting fatty acids, and of course, the microbiome, what we think of a lot of times, is the short-chain fatty acids that are produced by bacteria. And we worry about some of the bad bacteria like Clostridia producing propionic acid, which has been linked to autism symptoms in animal model. And we have some evidence that it could be disturbing metabolism too. So there is that suggests that kind of thought that, well, if you have bad bacteria and you have bad short-chain fatty acids, could carnitine increase that? Maybe, but I think that suggests that you got some work to do on your gut also to really clean that up. But carnitine in general is very effective. What I have found is that sometimes increasing carnitine very slowly, can be very helpful. Because again, one of the other issues is that carnitine does scavenge all of these toxic organic acids that are in your body. And if they have been hiding in cells and you give carnitine and you start mobilizing it, you're going to have to get rid of them. And so it may be that you mobilize things that you do have to get rid of, but you may overwhelm the body. So what you do is you give carnitine very slowly. You start at a low dose and you increase very slowly. But in general, I think the benefits of carnitine really far outweigh any of the drawbacks. You just have to know how to prescribe it and use it. And on that note, there's an open label trial that showed the advantage of using carnitine in autism from Baylor, and they used mega high doses of carnitine, I believe up to 6 grams, so 6,000 milligrams. I believe it was 150 milligrams per kilogram, which is a very high dose. And of course, the side effect that we see sometimes from carnitine is it loosens the stools, which can be an added advantage for some kiddos. and a lot of the GI guys use it to actually loosen the stools for kids with constipation. And so the major side effect, all they saw with really high doses was loose stools in some kiddos. So in general, it's very safe, and if you have too much of it, it'll come out in the stool, and you can back off on it. So, again, I think that's why it's important, and usually in the beginning of my lectures, I always talk about the fact that what we're talking about is off-label use of, these treatments. And whenever you use something off-label, you want to make sure that you use it in conjunction with somebody that has experience and can guide you in treatment.
Okay. So just to switch gears a little bit, I do want to spend some time talking about irregularities in the folate pathway. And so can you just share a little bit about why the folate pathway is important, what you see in autism, and what sort of factors contribute to a dysregulated folate pathway? We know that you've done quite a bit of research on the folate receptor alpha autoantibodies. And so that's one thing that I'm hoping that you can share a little bit about. And then also different genetic polymorphisms that may affect just our folate pathway and how our body takes up folate.
Yeah. So definitely the big player I'd say in the folate pathway is the folate receptor alpha autoantibody. I think that some of the changes in the folate pathway, the polymorphisms, at least in my opinion, I don't know that we really know enough about them and how they affect things to say how that would, if one or another is particularly important. And I'll tell you why in a second. But I do recommend that that children with autism take reduced folates as opposed to folic acid. And so one of the important nuances of folates is that our food is fortified with folate because it's so important, but it's fortified with folic acid, which is the oxidized form of folate, and some of us cannot metabolize it, so we can build up unmetabolized folate. The reason why we do that is because we can fortify our food with it because it's really cheap and it's synthetic. However, many individuals have problems actually reducing the folate into and getting it into the folate cycle. And we know that individuals have polymorphisms in something called the dihydrofolate reductase, which is kind of in the middle on the top here, which reduces the folate to get it into the folate pathway. Once we're in the folate pathway, we can see the folate pathway is connected to many different important parts of the systems in our body. On this diagram over to the right, we can see some of what we've been talking about. That is methylation metabolism. Methylation is important because it produces methyl precursors, methyl donors, which are involved in what we call epigenetic regulation. That is, it turns genes on and off. So that may be a reason many times that you may not have a mutation in a gene, but that gene may not be working because it may not be turned on through methylation. So we can't many times regulate as well if we don't have the right precursors. So methylation metabolism is very important, and that's connected to redox metabolism and production of glutathione, which regulates oxidative stress. Then we can look on the other side of the folate pathway to see where the arrows go, and they go into making something called purines. And what are purines? Well, purines are building blocks. We usually know them as the building blocks for RNA and DNA. So it's important to have them because you're always repairing your DNA and you're making RNA. And if you don't have the right precursors, you don't make it that well. Sometimes you can get errors in the code. So it's important to have purines as those precursors. But also importantly, one of the purines is adenosine. Adenosine is the building block of ATP, that really important energy molecule we were talking about. So if you don't have enough adenosine, you can't make the ATP that you need. In addition, there's another purine called guanfacine, and which makes GTP. GTP is the precursor to BH4, and BH4 is important for other biochemical systems that make all of your monoamine neurotransmitters. And those include serotonin, melatonin, dopamine, and norepinephrine. So essentially, folate really is the precursor and important for making all of those important neurotransmitters. It's also important for regulating oxidative nitric oxide and producing nitric oxide, and also phenylalanine. So folate is extremely important for making all of these important molecules in the body. So how do we regulate these folate pathways? Where are the blocks? Well, one of the things that people talk about a lot is the MTHFR, the methyl tetrahydrofolate reductase, which is an enzyme that helps this folate pathway function. And some people have what we call polymorphisms, where this may work better or worse, but it's still working. One of the great things, and we'll talk about the use of Leucovora and calcium or folinic acid to treat these disorders, is that your gut will transform half of folinic acid into methylfolate, which comes in on the other side of the MTHFR. So if you give leucovorin, you can get, or folinic acid, you actually have folate that comes into multiple parts of the folate pathway, and can be therapeutic in that way. So one of the big problems we find in the folate pathway is that folate isn't getting into the brain. And we found this back in 2012 or so, when we realized that some kiddos with something called cerebral folate deficiency, that is having low folate in their brain, were reported to have autism symptoms. So what we did is we asked, well, how many kids in our clinic may have this abnormality? And the reason for this abnormality is that there is an antibody, and let me see if I have a picture of this. Yeah. So we find that there's one of two antibodies. There's an antibody that is called the binding antibody that binds to something called the folate receptor alpha. And the folate receptor alpha actually takes folate from the blood and brings it into the cerebral spinal fluid and into the brain. And then there's the blocking antibody that blocks the ability of folate to actually bind onto this folate receptor alpha. So if you have one of these two antibodies, we find that folate isn't being transported from the blood into the brain very well. There's an alternative mechanism for getting folate into the brain, and that's called the reduced folate carrier, which we can see in the center there. Except that the reduced folate carrier, you see it's kind of smaller than the folate receptor alpha because it doesn't like folate as much. So you really have to give high doses of folate to force it through the reduced folate carrier. And the reduced folate carrier only likes reduced folates. So you have to give a reduced folate like folinic acid or methylfolate or such. Some of the other polymorphisms we see related to autism are polymorphisms in the reduced folate carrier. So there may be some kiddos that have a double whammy where they have the folate autoantibody and they have a polymorphism in the reduced folate carrier. So they have hard times getting folate into the brain. So in any case, what we did is we asked the question, well, how many kids in our clinic have these antibodies that prevent folate from getting into the brain that we're seeing with autism? We had no idea. We just knew that kids that with the antibodies and low folate, some of them had autism symptoms. So we actually, in our clinic, myself and Dr. Rosignol, we had 93 patients, and we found that about 60% of them had the blocking antibody, and about 50% of them had the binding antibody, or about 75% of them had one of the two antibodies that block the ability of folate to get into the nervous system. We then treated these kiddos that were positive with folinic acid, high-dose folinic acid, and we then, in about 4 months, asked the parents if their children got worse or better in certain areas. And we compared this to a waitlist control group. And what we found is that there were significant improvements in receptive language, expressive language, verbal communication, stereotype behavior, and attention in those kiddos that took the high-dose folinic acid. We then, that was an open label trial, so everybody knew they were getting the treatment. So then we did a double-blind placebo-controlled trial to ask, under carefully controlled conditions, do we see the same thing? And we looked primarily at verbal communication or language as our outcome measure, and we found that in 12 weeks, those kiddos that took folinic acid, there was a significant and marked improvement in their language. We also looked to see if the folate autoantibody could predict who were responders, and we found that yes, in fact, that it was. We were also able to calculate how much 3 months of treatment with folinic acid, how much equivalent speech therapy you would need to improve language that much. And we found that it would be about 177 hours or about $7,000 of speech therapy versus $300 of folinic acid. So not only did it give a benefit, but it potentially is cost saving. Many people also, we had some reports that initially when kiddos went onto leucovorin or folinic acid in the beginning, that there was some hyperexcitability and hyperactivity, which then got better several weeks into taking the medication. And we hear about this in the clinic. So one of the things we did look at is the reported adverse effect of agitation or excitement during the trial in both the kiddos that were treated with leucovorin and those that were treated with placebo. And what we found was very interesting, that yes, about 20% of the kiddos treated with leucovorin reported that that there was some hyper excitability during that time point, but it wasn't really significantly different than placebo. And at 6 weeks, so at both 3 and 6 weeks, they reported that. But at 9 weeks, there was a precipitous drop in hyperexcitability only in the kiddos that were treated with leucovorin. And we saw in our study that it does seem to improve hyperactivity. So this may be something that we see early on, and sometimes we have to increase it slowly. But in our study, it suggests that it's no different than placebo. One of the other things that's very important, which I'll just mention briefly, is these same transporters that are on the brain that prevent that can be blocked by this antibody for getting folate into the brain. Those same transporters are on also the placenta. And that about half of the mothers of kiddos that have the antibody also have the antibody. And we do suspect that it could have a role prenatally with getting folate to the fetus and interfere with development. And so that's why I always recommend women that are pregnant to take reduced folates. So if they have any polymorphisms or any type of blockages in the folate pathway, they have better chance of folate getting to the fetus.
Dr. Frye, I just want to be respectful of your time. I know that we're approaching 3 o'clock here. Do you have any other obligations after our talk here in case we go over? I just want to be respectful of your time.
Sure, I do, but I can spend a couple extra minutes.
Okay, so a couple questions that have been coming up is what do you see as far as using leucovorin versus L-methylfolate? Do you recommend one over the other? And what dose do you usually recommend that we could share with our providers, for example, who may not be as familiar with this protocol? Where do we start with treatment as far as leucovorin goes?
Yeah, so we use leucovorin for 2 reasons. One is, as I mentioned, and when I was showing this slide, that half of leucovorin is metabolized to 5-methyltetrahydrofolate. So you get the best of both worlds. If you have a blockage in the MTHFR, you get folate on both sides of it, which is really important. And I think that leucovorin is very important in getting to folate in before MTHFR is very important, because as you can see in this diagram, you have the shunting to purines, which are very important for mitochondrial metabolism and for producing neurotransmitters. I guess the question was, what is the dosing? The dosing that we've used in the clinical trials of leucovorin is 2 milligrams per kilogram per day, divided in twice-a-day dosing, with a maximum of 50 milligrams. So usually a maximum of 25 milligrams twice a day. The pharmacopoeia says that is the maximal amount that you can absorb, 25 milligrams at one time. There are children and people, especially older kiddos, that I will say respond to higher doses, but we take that in a case-by-case basis. The other reason why we use leucovorin is that it's been used for decades in oncology, and so we know its dosing and we know its safety profile. So we have a lot of information about it.
And just looking at some questions from your audience here, a lot of folks have undergone the FRAT test or are interested in getting the FRAT test. Can you just quickly describe the difference between the binding and the blocking antibodies? And if you recommend that families pursue this test?
And what was the, I'm sorry, what was the end of that?
That's okay. Just wondering if you recommend that families pursue this test in your clinical practice.
So yes. So in addition, to all those things I described as, the screening labs for mitochondrial dysfunction, when I see a patient, I also do the FRAT test. I recommend that at least patients get the test. It's usually not covered by insurance, so it does have some out-of-pocket cost. The studies that we've found is that the FRAT, the folate autoantibody, seems to be predictive of those individuals that will respond to leucovorin. So I use it as a biomarker of kiddos that will respond to this treatment. So yes, there may be kiddos without the FRAT, so that are not positive, that respond to, but at least in our studies, they seem they probably respond much less. And so there may be other treatments to pursue, if that's the case. So I always use the FRAT test. I also do that because if you treat without doing the FRAT test just empirically, and you don't see a response, then you're left with the question of, well, was I, supposed to treat in the first place or not? So I always like to have more information.
Okay, and is this something that other providers can order just in their clinical practice? How do we access the test?
So Iliad Neurosciences is the company that does it, and you just order it online, and I believe they send you a kit, and it's easy to do. I believe you don't really even need the kit. It's I think a standard red top tube. Only takes a couple milliliters of blood, but they will send you a kit if you want. So that's something that people can find very easily online. They can type in FRAT. I think usually it comes up if you put in Google FRAT or Iliad Neurosciences.
Okay. And families are asking. Oh, I'm sorry. Go ahead, Monica.
One question that some parents had was sometimes the test comes back as indeterminate, and the recommendation is to repeat it again. Would that in that case, would you recommend to go ahead and treat empirically and then repeat the test in a few months?
Yeah, it depends on why it's come back that way. Now, the thing that we don't know about, which is kind of a third, and sometimes is why it comes back indeterminate. We know about these blocking and binding antibodies. Again, the blocking antibody blocks folate from binding onto the folate receptor alpha. The binding antibody binds to the folate receptor alpha and kind of messes up the way that it works. But then there's another finding that is sometimes reported called the soluble folate receptor. And this essentially means that these folate receptors that you see here that are bound onto the cell, for some reason, have broken off and are floating around the blood. And although we haven't studied that, clinically, I interpret that as bad. Yeah, I think if the folate receptor is not where it's supposed to be and it's floating around the blood rather than being on the epithelial cell, I consider that bad and dysfunctional, and I would treat.
Dr. Frye, does Leucovora need to be given with vitamin B12? What is your opinion on that?
So I give when I give B12, I give B12 with folinic acid. I combine them, and it's a different reason. I think the opposite. I think B12 has to be given with folate in order for it to be optimally effective. So when I give B12, I use the dual shot by Lee Silsby because I find that it has less side effects and it's more effective to give those two together. But that's a lower dose of folinic acid. So if you're treating cerebral folate deficiency or a transport mechanism problem, you want to use higher doses of folate.
Okay. Thank you so much. Dr. Frye, one of the families that are asking, how can they see you in your clinic? How can they get an appointment with you?
Sure, you can contact the clinic, or you can email me, and I can connect you with Nicole, who's my coordinator, who could set up a clinical appointment.
And we also wanted to know if we might be able to have access to your slides.
Sure. Yeah, I can give you. This is a slide deck from a talk I gave recently, but yeah, you're welcome to have them.
Great.
All right. Well, Dr. Frye, thank you so much for your time. I am sure that your conversation with us today answered many questions for many families. Are there any closing remarks that you want to make?
No, I'm just, happy that that you guys invited me, and I hope that it helps, some kiddos.
Absolutely. Yes. Thank you again so much for your
Yeah.
time.
My pleasure.
Yes. Have a good rest of your
All right.
day. Thank you.